Welcome back, Bio30 Prep. This is your last lecture for Unit 1, talking about the cell structures and organelles, and we are going to be talking about the cell membrane and transport through the cell membrane. Again, your textbook, you can read pages 156 to 159. By the end of this lecture, you will be able to describe the structure and function of a cell membrane, compare passive transport and active transport across the cell membrane in terms of energy requirements, describe diffusion and osmosis, describe endocytosis and exocytosis. So here we go. Here is a picture of a cell membrane. So this is the membrane that surrounds the entire cell. And if we just take a look at this picture, notice that it says here we have outside the cell, over here is inside the cell, and so you have to imagine the cell membrane kind of extending all the way around a cell. So this is a thin cross section of just the membrane around the cell. Every cell has one. It is selectively permeable. In other words, some substances can travel through it, others cannot. And it's a thin bilayer of phospholipids and protein molecules. I won't get into the definition of those things right away. But if you take a look, the phospholipid bilayer are these kind of spheres with little tails on them. And the proteins are these big blue structures that either travel all the way through the cell membrane, or some of them are embedded either on the inside or the outside, depending on what their purpose is. So they're represented by great big blue blobs, the proteins are. The main purpose of the cell membrane is it separates the cell's interior from its surrounding environment, and it controls what substances get to move into and out of the cell. The cell surface, those proteins, their main responsibility is controlling the movement of particles into and out of the cell. They also catalyze chemical reactions. The plasma membrane is only about 10 nanometers thick. A nanometer is 1 nanometer is 1 times 10 to the negative 9 meters, or 1 millionth of a millimeter. So if you took a millimeter and cut it up into a million little pieces, go find yourself a ruler and just take a look at how big a millimeter is. If you were to divide it up into a million separate pieces, the distance between two of those lines would be the thickness of a cell membrane. It's only faintly noticeable with an electron microscope, so they're not very thick. Here's a bigger picture of the same thing on the previous slide. 2 layers of phospholipids called the phospholipid bilayer, and with a distinctive head and tail shape of the phospholipid molecule. So we're going to actually find out that the little round part, this is the phosphate, and the tail here, these are the lipids. And lipid is another word for fat. So these are fatty little tails on the end of phosphate heads, and in terms of the chemistry, a phosphate ion is this thing here. It's 1 phosphorus attached to 4 oxygens, and it actually has extra electrons with a 3 negative charge. So that's what a phosphate molecule is. Inside the cell, notice that the cytoskeleton is supported by the cell membrane. So these strands here, these would be microfilaments used to support the cell. When we look at the membrane itself, it contains protein molecules. Their purpose is to act as gatekeepers. They allow compounds to either enter the cell, or they react with hormones that have been sent from other areas of the body. So for example, when hormones leave our brain, they travel through a circulatory system and will target different glands. When they get to the cells in that gland, the proteins on those cells interact with the hormone that the brain has sent. There's 2 types: carrier and channel proteins can move things in and out. Glycoproteins and glycolipids, so known as glyco that means sugar. So here we have a sugary protein and a sugary fat. They kind of act as markers, identifying markers to let other cells know what kind of cell this is or where this cell came from. So they're kind of like a signature. The other thing that's on a cell membrane, a plasma membrane, are phospholipids. We already kind of went over that, where we have the phosphate ion on the head, so water-loving heads, and hydrophobic or water-hating tails. And this is what keeps the phospholipid bilayer together. If we just go back one slide here. So outside the cell, there's lots and lots of water molecules. Inside the cell, again, lots and lots of water molecules. The lipid tails are always being pushed away from the water, so as water comes up against the cell membrane, it pushes these tails on the inside into the inside of the bilayer. Same thing on this side. So those phospholipids are actually forced into their shape by the presence of water because those tails are trying to get away. Think about oil always rising to the surface of water. Well, if there's no surface for it to rise to, it binds itself in a layer. So we'll just get back through this slide here. And so, these lipid molecules are mobile, so they aren't in a fixed place. They kind of slide along each other, kind of like those little phosphate heads roll along each other. So the membrane itself is a fluid structure. When things want to get into the membrane, there's two ways that the cell can acquire nutrients and get rid of waste. Remember, every time nutrients are taken in, after they're used, waste products are formed, and those waste products have to make their way back out of the cell. So for moving across the membrane, there's two ways to do it. One is passive transport. It requires no energy. So without energy from the cell. And there's three types. We'll talk about all three types in this lecture. There's diffusion, osmosis, and facilitated diffusion. The other choices, and this is especially true when you're talking about things like glucose or amino acids. Cells need these things for energy and for functions. Without them, the cells will stop to work. They'll die. So these substances are so important that waiting for things to come in passively isn't a good idea. So there's active transport. It's a way to move stuff across the membrane by spending cell energy. Other choices include endocytosis and exocytosis. And they require energy, but what happens is these are for large molecules. They're too big for protein transport. So there's got to be a third method of transportation through a cell membrane. Diffusion, I like to call it shopping on a Tuesday, and here's why. If you imagine going to West Edmonton Mall on a Tuesday in, say, the middle of, February. Right, so Christmas is done, nobody has any money. If you were to go shopping at West Edmonton Mall on a Tuesday afternoon in February, chances are you could go wherever you wanted, whenever you wanted, and spend very little energy. You could walk around with your eyes closed and eventually bump into the store you're planning to get to. So it's called Brownian motion, and basically it's the idea that molecules move randomly through or past each other, caused by molecular collision. So every time a molecule bumps into another molecule, it changes its direction, and the direction is, for the most part, random. We can't predict exactly how one specific molecule is going to move through a substance. But the general result is that molecules move from an area of high concentration to an area of low concentration, or the fancy word for it is with a concentration gradient. And if you think about when you have a cup of tea, if you put that tea bag into a cup of hot water, initially the water is mostly just water, and all of the tea is inside of the tea bag. When you put the tea bag into the tea starts to diffuse out of the bag and into the water. If you leave it long enough, then the tea evenly distributes itself throughout the water. So it moves from an area of high concentration, this would be inside the bag, to an area of low concentration, which would be into the cup. And if you leave it long enough, it reaches a point where the concentration of tea throughout the entire cup is exactly the same. The rate of diffusion is affected by a bunch of things, and most of these are fairly intuitive. Affected by temperature. If the temperature goes up, then Brownian motion increases. Concentration. As concentration goes up, the rate of diffusion increases. And as pressure goes up, the rate of diffusion increases as well. All of these things, when they go up, they increase the rate at which molecules move through a space. Now, one of the problems with diffusion, or with passive transport, is at best we can hope for equality. At best you can hope for is the concentration of tea to be the same inside the teacup as it is inside the tea bag. And that's the best you can hope for all of those tea molecules that are dissolved in the water itself. So the best you can hope for is an equilibrium situation. In our bodies, oxygen and carbon dioxide, for the most part, move through cells in this fashion. In animal cells, CO2 moves into the cell, and CO2 moves out of the cell, and it's pretty much by diffusion. Mostly because, if we think about it here, when cells are working, the concentration is high inside cells of CO2. So the CO2 concentration inside of a cell is very high, so it naturally moves out of the cell and is going to move into our circulatory system where we can exhale it eventually. The opposite is true of oxygen. Cells are always using oxygen, so the concentration is low. And if the concentration of oxygen inside the cell is low, then oxygen from outside that cell will naturally diffuse into it just by random motion to equalize concentrations. Facilitated diffusion is kind of the same idea. Often it could be because these molecules are too big to fit through a plasma membrane, or it could be just a way of speeding up diffusion, but this is diffusion aided by membrane-bound proteins. Two types. There are channel proteins, and this would be one here. There's a channel protein, and really all it is a space that molecules can go through. There are also carrier proteins. Carrier proteins can grab specific molecules and shunt them through the cell membrane. So, the important part with facilitated diffusion is it's still moving solute, and solute would be the substance dissolved in water. For us, we're talking mostly about ions and nutrients that are dissolved in the water or in the cells that need to be carried in. So this is still movement with the concentration gradient. Now, if molecules are too big to fit through the cell membrane, there's still a force that wants to equalize concentration. So osmosis is a force where water diffuses through a selectively permeable membrane instead of the solute itself. So if a solute molecule can't move through the membrane, then water will, in order for the solution to reach equilibrium. So the idea is substances naturally try to get to a situation where it's almost as if the membrane didn't exist. They want the concentration to be the same throughout the entire area, just by Brownian motion. So if the solute can't move through the membrane, water will, and until the concentration of the solute outside the cell is the same as the concentration of solute inside the cell. Here's an example of what osmosis can do. There's some terms here: isotonic, hypotonic, and hypertonic. Isotonic means that the concentration of solute inside and outside the cell is equal. So here it's always about the concentration of solute. A hypotonic solution is when the concentration outside the cell is less than the concentration of the solute inside the cell. So here we have a hypotonic solution. If the concentration outside is less than the concentration inside, water wants to move in to the cell to equalize the concentrations. The concentration of the solute outside the cell is high, so water moves into the cell and it causes the cell to increase in size. Here we have a plant cell and we can see water moving inside of it for a hypotonic solution. For a hypertonic solution, this is a solution where the salt concentrations, or the solute concentration, is higher outside the cell than it is inside. So here we have lots of salt, or lots of solute we'll call it. So water leaves the cell in order to dilute the concentration of that solute. So lots of solute, little solute. You end up losing that water out to the outside of the cell. Turgor pressure is a word used for the pressure exerted by water against the cell membrane and cell walls of a cell. So if we take a look at this first picture, here turgor pressure is low because that cell membrane is being sucked in inside the cell. In the middle picture, turgor pressure would be average. The last picture here shows a high turgor pressure because as water enters the cell, it starts to push that cell membrane up against the cell wall and the cell wall outward. So here, the turgor pressure would be high. Here's pretty much the same kind of idea, but here we're showing some red blood cells and what happens to the cells. So we showed it in the last picture. We'll show it again here. Cells in isotonic solution undergo no change. The water going into the cell is equal to the water leaving the cell. Cells in hypotonic solutions expand as water moves into the cell to decrease solute concentrations. So as those cells expand, it's possible, and we'll talk about this a little bit when we talk about circulation, but it's possible to actually burst cells by forcing water into them. And lastly, in hypertonic solutions, cells shrink as water moves out to increase the solute concentration inside that cell. So now we've covered passive transport, we're going to take a look at active transport, and this is what I call shopping on Boxing Day. If you want to get somewhere in West Edmonton Mall on Boxing Day, you have no choice but to push and shove and move to that store that has the best deal. Same idea with cells. Cells want specific nutrients and they want a lot of them for cell functions. The only way to get them is to go out and grab them because the concentration of those substances is always going to be lower outside the cell than inside the cell. So it's the transport of molecules against a concentration gradient. So molecules are going from an area of low concentration to an area of high concentration. And that's completely opposite of what diffusion does. So the molecules are going this way. They're going from low concentration into an area of high concentration, and so that takes energy to do. In terms of the human body, it's necessary for nerve conduction, muscle contractions, kidney function, so all of these things require a use of energy to increase solute concentrations inside the cell. When we're sleeping, and this is one of the reasons why sleep is important, 30 to 40% of your energy budget is spent while you're sleeping. It's engaged in active transport, and what we're going to find out is the human body is a little bit like a mousetrap. And what I mean by that is, when I talk about mousetraps, I'm not talking about the nice, the animal-friendly mousetraps. I'm talking about those ones that are just a board with that little piece of metal on a spring. And now, the way a mousetrap works is you spend all of the energy pulling that metal hinge back and locking it in place. That's when you spend the energy. When the mouse actually comes to eat the cheese and the trap is sprung, that doesn't take any energy at all. All of the energy is spent beforehand to kind of get the trap ready for when a mouse shows up. And our body acts a lot in the same way. For example, for muscle contractions, the energy isn't spent when you move a muscle. The energy is spent while the muscle is resting. And that muscle kind of gets spring-loaded, ready for a contraction. Same thing with nerves. Your nerve cells, they set themselves up before they need to do anything. So before you sense anything, with, say, a sense of touch or a sense of sight, all of your nerve cells are set and ready to go. They spend the energy to get ready to sense something. When you actually experience sensation, it doesn't take any energy to make that nerve work. So our body acts like a mousetrap in terms of storing up energy. Two other forms of transport. Sometimes substances are too big to move into and out of just a protein. So it's a process by which cells engulf molecules that are too large to pass through the cell membrane. These substances wouldn't fit through, for example, this would be large proteins. So these large proteins, they won't fit through a cell wall. So what happens is they're engulfed by a section of membrane and either pushed out of the cell or sucked into the cell, surrounded by that membrane. So really what happens is the cytoplasm, and here we have an example of endocytosis, these particles here are too big to fit through the cell membrane, so the cell membrane makes a pocket. And in that pocket, it's going to pinch that pocket off, and the vesicle is going to travel into the cell. And by doing that, it carries those large particles into the cell. If there's a waste product that a cell is trying to get rid of, well, then the reverse happens. Here we have a vesicle. It starts closed off, and then as soon as it meets the cell wall, it opens up, and it pushes all of those substances back out into the extracellular fluid, or really this is just fluid outside of each cell, or the space between cells. An example of these is the lysosomes we talked about in the previous lecture. Stored inside of them are digestive enzymes that can be used to break down other particles. In terms of endocytosis, there's two types. They're the same thing, just what they're taking into the cell. So for endo means in. So there's penocytosis and phagocytosis. Penocytosis is the absorption of liquid droplets. Phagocytosis is the engulfing of solid particles. It can be used for cells to obtain food. It's also used in our immune system by white blood cells. That's one of the ways they ingest foreign microbes and then unleash digestive enzymes on them to tear them apart. So here's a summary of what you should get from this cell membrane lecture. Probably one of the most important lectures for this first unit. So getting down the ideas of diffusion and osmosis, knowing what they mean, and especially active transport. These things are really important to get through. So for the plasma membrane, we just had this idea that it's a phospholipid bilayer. It contains these embedded proteins that are involved in transporting substances into and out of a cell. So the purpose of the cell membrane is to regulate the movement of substances into and out of the cell. And then these are the methods here, type of membrane transport and characteristics. If we just were to group these into two groups, the first three require no energy. Different ways for things to move in and out of the cells. No energy is required by the cell. There's no energy output by the cell. The last three all require energy. and they're just different types of, these all require energy. So energy is needed for all of them. The difference between them is active transport by itself is for small molecules. Endocytosis and exocytosis are for large molecules. And there we have it. That is the end of your first unit. When you're ready to head on to Unit 2, I will see you.